Welcome, everyone. We're just letting a few more folks trickle in here. We're going to go ahead and get started in just a couple of minutes. But first and foremost, just want to say thank you so much to all of you for joining. We're so excited to have you all here. And we'll get rolling with the opening session and then finally getting into the questions in just a couple of minutes. All right, everyone, we are going to go ahead and get started rolling into our introductions. Welcome again. We are so excited to be kicking off this first panel of the Nucleate Inside Aging series with all of you today, focused on the landscape of aging biology and longevity biotech. My name is Sean Simonini, and I'm one of the co-managing directors of the Inside Aging series. I just want to quickly say thank you to all of you for joining us. Today's panel wouldn't have been possible without the help of the amazing Path team at Nucleate. Shout out to Andrea and Trudy who are here, as well as everyone in HQ who supported this initiative from the very start, including our co-president, Zach Kogan. We have two great moderators for today's discussion, Zatvik and Sierra, who will introduce themselves in a little bit. And we're incredibly grateful to our speakers, Dr. Eric Verdin and Dr. Martin Borsch-Jensen, for taking the time out of their very busy schedules to join us. They're true visionaries in the space, and we're honored to be able to hear from them today. Today's panel and the several to come are ultimately going to be aimed at helping the research and biotech community get a better understanding of what aging biology is, its immense potential to improve human health span, and most importantly, how you can get involved in building a future where we all live healthier for longer. So whether you're a graduate student, an investor, an undergraduate, or anywhere else in your journey, we hope that there's a lot that you can gain from this conversation in the ones to come, and we hope that this is another step towards a future where we can all live healthier. And so I want to go ahead and make a couple of quick announcements before we get started and I transition it over to the moderators. We do have several other panels coming up after this with some other amazing speakers. Obviously, today we're talking about the landscape of aging biology, which is our introductory session for the panel. But on March 3rd, we'll be talking about the path to the clinic for aging, focusing specifically on the translatability of longevity and aging biology interventions. Then in April, we'll be focusing on aging clocks, omics, and other biomarkers of aging that are helping to revolutionize how we think about the field. And then in May, we're going to be talking specifically about the venture capital side, something that I know is going to be of incredible interest to all of our members of the global Nucleate audience and beyond, talking specifically about startups as well as venture capital with some great friends from Age One and other venture capital spaces. And so I also want to note that we will be putting out resource guides to accompany all of these panels that we're running. We'll be posting the first one in the near future. Just keep an eye out on the Nucleate socials for that, specifically our LinkedIn page, and for the announcements and registration pages for all of these upcoming panels. Keep an eye both on the LinkedIn and then also on our Luma Global Events calendar page. Everything will be posted there so you can sign up and make sure you get the Zoom link so you can access more great content in the aging biology space. And so at this point, it's an absolute honor to be able to introduce the speakers that we have for today's panel. Dr. Eric Verdin is the President and Chief Executive Officer of the Buck Institute for Research on Aging. A native of Belgium, Dr. Verdin received his doctorate of medicine from the University of Liège and completed additional clinical and research training at Harvard Medical School. He's held faculty positions at the University of Brussels, the NIH, and the Piquet Institute for Medical Research. Dr. Verdin is also a professor of medicine at the University of California, San Francisco. We're also glad to be joined by Dr. Martin Borsch-Jensen, who is the founder and CSO of Gordian Biotechnology, the first in vivo therapeutic screening platform to radically improve drug development for complex diseases of aging. His focus is on developing technologies to interrogate the full network of aging processes inside of living organisms. This includes multiplex measurement of complex states, interpretable perturbations, and context-aware data models. Additionally, he leads Norm Group, which is a nonprofit focused on supporting and conducting research that helps people live healthier for longer. We have an incredible conversation ahead of us. And so without further ado, I'm going to go ahead now and pass it off to Satvik and Sierra to briefly introduce themselves and then dive us into our question and answer session. Satvik, I'll pass it over to you.
All right. Hey, everyone. Thanks so much for coming. My name is Sathik. I'm a student at Harvard, and I also work for H1, which is a venture fund that backs biotechs that are targeting the biology of aging to treat, prevent, and reverse disease. Super excited for today's session, and I'll turn it over to my co-moderator, Sierra.
Hi, everyone. Thanks for being here. My name is Sierra Laurie. I am a PhD student, Eric's lab, actually, in my second year. And my background is Stanford bioengineering. And yeah, excited for you all to be here today and for this fun discussion.
All right. So with no further ado, we'll get into the questions. So I want to start with a question to both of you. I'd love to hear your personal definition for aging or any mental models that you have for explaining this really complex process. So how about we start with Eric? Yeah.
All right, very good. Well, first, congratulations for having this event, and thank you for having me, and I look forward to the discussion. How do I think about aging? I think about it as there are a number of forces that are causing aging over which we have no control and most likely will never have a control, which is think about gravity, the fact that we're living in a highly oxidative environment by breathing oxygen. Think about gamma rays coming from outer space. Think about ultraviolet. Can wear sunscreen. That's one way where we can mitigate this. But so there are a number of forces that are really driving the aging process. Now, what counter to this is a whole series of adaptive mechanisms, protective mechanisms that we have developed that allow us to repair the damage that's caused by gamma rays and so on. So think about DNA repair, protein folding, all of the mechanisms that maintain the integrity of our biology. The way I think about aging is these repair mechanisms are pretty perfectly balanced with the damage for the first, between age 20 and 30. And it's been estimated that if your aging rate would happen, we continue at that rate, we would all live to 1,000 years old. Unfortunately, that's not the case. So that as we age, these repair mechanisms, so that the insults stay pretty much constant, but what happens is the repair mechanisms slowly degrade. And so that leads to the progressive, very slow accumulation of damage. So the aging process is a reflection of this progressive accumulation of DNA damage. At some point, the damage targets the repair pathways, which accelerates the whole process. So one really important thing to think about aging is the fact that it's a slowly exponential process, extremely slow in the early parts of life, but slowly accelerating and culminating, unfortunately, in death.
Yeah, super interesting. I think the exponential piece reflects the incidence of age-related diseases over time. So I'll turn over to Martin for his concept.
Yeah, I guess, I don't disagree with what Eric is saying, so it's more like a supplement. I think this way of okay, damage in, damage out, it's sort of like a differential equation, like linear math thing. My one way that I think aging works, but I can't prove it yet, is if you think of an organization, a government, a company, whatever, the body is just like an organization of specialized cells that are supposed to do stuff in response to each other in the right way. And you see a lot of diseases is not that, right? It's like the immune cells are overactive, and then you cause fibrosis by having persistent inflammatory signaling or something like that. And I think that there's in addition to what Eric said, I think of a sort of coordination aspect to aging, where you have a complex system that needs to be aligned. And when you lose that alignment, then you can also get an sort of exponential loss of function because you have feedback loops, right? And so biology is full of feedback loops all over the place, right? And so when you get out of whack, when you don't have a sort of governing, if you had a company with or a Buck Institute with no CEO, right? And everyone's just like going around doing random stuff and it sort of falls apart. So I think this sort of like systems aging, coordination, loss of coordination between specialized function is something that we see when you look at sort of, you affect this organ and the other organs get affected, or you can transplant, senescence and young organs and parabiosis and so forth. But that's not like heavily studied because it's much harder than studying the cellular aspects of aging. It's sort of one or maybe two levels of sort of complex organization up from cellular aging, and you can't just grow it in a lab, right? Or you can, it's a whole animal, so it's like a lot of logistics. Yeah, so that's my add-on to without disagreeing with anything.
Thanks, Martin. Now turning it over to Dr. Verdon again and focusing on one specific area of aging in this model that you've talked about. How specifically does immune aging contribute to aging-related diseases, and how do you feel about it as a point of intervention?
Well, I'm glad we're having this question because it is actually, as you know, Sierra, a big aspect of my lab is actually working on. As I play two roles. I'm the head of the BAC, but I also run a lab that focuses on the interface between immune aging, metabolism, and epigenetics. And the way I think about aging in the immune system, so when I joined the BAC about 8 years ago, we decided to initiate a new program focused on human immune aging, and realizing that this is an area that was and still grossly understudied. Some of the factors that really led us to study human immune aging are, first, it is one of the most accessible organs that we have in terms, you just need to draw blood and you can pretty get the whole complexity in a test tube. So that's one aspect. It's also amenable to intervention. If you think about the fact that we can extract T cells, we can reinfuse them. This is applied to biology and medicine right now in the whole context of CAR T cells. So these were two variables that I thought were important for us as a lab to really focus on having an accessible and human system that we could study. The reason that the reason that it emerged actually later was the emerging evidence that the immune system can be a dominant factor in the aging process. There are two papers that were published in the last 5 years, one of them by Laura Niederhofer, showing that aging in the immune system, at least in mice, can be dominant to organismal aging, meaning if you induce a lesion in the mouse immune system, this was done in these two papers by either disabling in the immune system specific manner, a gene that controls DNA repair, ERCC1, or damaging a gene that controls mitochondrial biogenesis, the transcription factor called TFAM. When you induce these lesions in the immune system using Krelok's type of mouse approach, you actually you induce secondary senescence. So first you induce senescence in the immune system, but also really interestingly, you induce secondary senescence in other organs. And so I think that speaks to a potentially driver role for the immune system in organismal aging. Obviously, this has not been studied in humans. This is what a large part of our research program are, measuring immune aging and now actually trying to identify and study the mechanism. So these were, for me, this is something that should be kept in mind. I'm not saying the immune system is the driver of aging, but it definitely has that potential that was illustrated by these papers.
Absolutely. And as a leader in the field, you're really pushing this immune aging space forward, and it'll be exciting to see what happens in the coming years.
Absolutely. If I can add one more thing, one thing that really struck me when I did a lot of work on immunology and HIV prior to moving to the aging field about 20 years ago. And one thing that always struck me is that if you go to an immunology meeting, almost no one talks about aging. And if you go to an aging meeting, almost no one talks about immunology. So, and I think one of the reasons is, as a field of research, immunology is probably, along with neurobiology, is probably one of the most advanced. There's enormous amount of studies on immunology and its complexity, its role in protection against viruses and so on. But there definitely is still to date a chasm between the two fields. And so that's where we've jumped in and trying to sort of lead the way and at least, along with many other colleagues, but really establish the role of the immune system in the aging process.
We look forward to seeing it unfold.
Yeah, we need more, you know how those Keystone meetings that have like metabolism and, liver disease or something. We need a lot more of those that are like aging plus X, where X is just all these diseases of aging, because they are these silos, like Eric's saying. So anyone on the call, you should organize one. You may have to get some tenured professor to co-organize it with you.
And as with any field, there are also hurdles. So moving over into the side that's more challenging. This is a question for both of you, and maybe Martin, you can start us off. In your opinion, what are the biggest challenges and roadblocks currently facing the field of aging biology?
Yeah, I think I don't know, this should be uncontroversial, right? Like, one, it's slow, and we don't have any actually validated biomarkers that have predictive power, right? Like, we have a bunch of methylation clocks, and nobody's like taken a clock and then said, look, we don't need to do a lifespan experiment because I've now proven that this actually predicts like the mouse lifespan or whatever, right? And so that's really important, both on the research straight, like a postdoc in aging takes longer, right? Like, bad idea. When I was doing mine, that was in fruit fly, so it's easy, but it's still 3 months per experiment, right? Just slow. And then obviously for like anything clinical trial kind of stuff, right? Like, that's a huge thing. So I think that's the most obvious. Then I would say secondarily, I mean, catch-22. We want to figure out how to measure aging. And so now we need a drug that we're sure works, but we don't have a drug that we're sure works, right? Like, we have good results for rapamycin and other things like in animals, right? But it's sort of like, you don't have it like here's 20 things that are really good and like 80 things that are really bad, and you can like really train these predictors, right? So there's sort of like a catch-22 there. And then I would say, just like from my own personal take, it's, if we go back to sort of like a lot of the parts of aging are connected, we don't have great tools to measure that. And so it's hard to say, like, if we think that, oh, actually, if the immune cells are in this state, but it's only some of the immune cells, then they can induce senescence in like other organs, right? Like, as Eric was alluding to. How do you measure, like, how does that work? How do you measure that whole sequence of events at once? Because a lot of this aging stuff only happens in vivo, right? Like, it's, that whole thing wouldn't apply if you're just talking about like a culture of senescent cells. And so how can you get those measurements that are sort of like happening over multiple different timescales? How can you get that tracked and therefore come up with like a cohesive system of like how the things affect each other? I think that would be my other, those things. And then just like, it's pretty small field. Like, I don't know how many people, 3,000 something. And it's like 0.5% of all biomedical research spend, right? So that's the like idiot answer of just like, you're working with limited resources. I do think that one is, obviously you're addressing that in part right now in the session, but also like, assume that that's the case and then figure out what's the high leverage single thing to solve.
So if I can jump in on the same point, I agree with everything that Martin just said. I think if I had to really, for me, I really think about the space in two big blocks. One is the basic understanding of the biology of aging, which is conducted mostly in animal model systems for a number of reasons, the ease of really modifying genes and so on. And human aging and how we're going to be translating all of these discoveries to humans. I mean, at the end of the day, at least for me, not for all colleagues, my goal is really to improve human health span and the longer term lifespan. So if I think about the biggest roadblock in the basic biology of aging is we are a bit, you probably have seen this cartoon of people, in a room with a scientist in a room with an elephant. One is holding the trunk, and the other one is holding a paw and a foot, and no one really has a clear understanding of that it is an elephant. So we are still at the stage in aging biology where we've defined what we call 10 hallmarks of aging, which are a set of abnormalities that characterize the aging process. And they are, they've been validated, they're important. However, it's a very fragmented understanding of what aging is. What we're lacking is sort of an integrated view of all these hallmarks interact with each other. Is there a hierarchy? When we've been very good at identifying drug targets, how do these targets synergize or antagonize each other? So there's a whole aspect of system biology, which the field is really moving in very aggressively. By the way, this is not something unique to aging. It is something for all of biology. We've moved from the study of single genes to really an integrated understanding. And the problem when we move into this highly complex data is our inability as based on our biology to really understand therefore the role of AI as a new tool, which has dramatically accelerated our ability to understand complex data sets. So I really view this as a big area of focus in the field. In terms of human biology, I think also people talk about this as translational research. I think about it as a different way of doing aging biology using another model system, that model system being humans. The limitation there is first, there's been much less work done on humans in terms of understanding how we age than in a mouse or C. elegans or Drosophila. And the second one is, as we identify potential interventions, how do we conduct these trials? No one is going to do a 30-year clinical trial. There's a clear other problem in humans is that if you're going to be proposing something, you better be careful, ensure that it's actually safe. There's nothing worse than to, test an intervention in someone that actually is going to hurt them or shorten their lives. And obviously, the slowness of the aging process in humans really calls for what Martin described as biomarkers of aging, which are really critical in predicting whatever the outcome is going to be of an intervention that you're about to try. So, and I think we see, I make the point that despite all the glamour and all the excitement in the aging field, we still don't have a single drug or a single a supplement or any interventions that we know reproducibly increases health span and lifespan in humans. We have lifestyle factors. We can talk about this hopefully later, that we know are associated with life, increased health span and lifespan, but it's a very different thing to conduct a clinical trial. Last but not least, if you think about lifestyle factors are enormous in terms of human health span and lifespan. Right now, we have mostly epidemiological studies. So I don't think about human aging as just finding a drug that's going to cure everything, but more really optimizing these lifestyle factors, which we know are major contributors to how we age. So understanding what is the best form of exercise, nutrition, testing those using the scientific method, and really being able to provide the population with some clear answers in terms of what are you supposed to do if you want to maximize your lifespan. I think I'm equally passionate by these as I am by drugs, because essentially those are available today, and the epidemiology points to their efficacy.
Just adding a quick question onto that, why do you think it hasn't been done so far? Is it mostly lack of data? Is it lack of project initiative in this area in terms of getting a complete and holistic view?
I think, like anything in science, we are always limited by technology. I would say that the clocks that emerged, about 10 to 15 years ago are amazing. They really have true predictive value. Although, the field right now is a proliferation of them, it's really hard to understand which one is going to be the key one. If you want to have a clock that works, it needs to be both predictive, but it also needs to be accurate, and it needs to be portable. You need to be able to do it in people. So there are a lot of variables. Right now, last I counted, there were close to 60 different clocks they all tell a different stories. Like according to them, I'm between 25 and 67 years old. So when you look at this, you realize, these are tools that are not ready for prime time. Some of them are being commercialized, but I've argued that they're not ready for prime time. And so we as scientists need to sort through all of this to really learn how do we use them as tool for clinical trials and also for global health.
That makes sense. Thank you.
Yeah, that's a great point. I think translation is hugely important. And we'll actually be having a session all about clinical trials and the path to clinic for aging in March to continue the discussion about endpoints and biomarkers and all these topics. But yeah, thanks so much for the insight. I think next, going deeper into this idea of translation and going from preclinical experiments to things that can work in humans, I want to go deeper into Martin's work with Gordian's platform. So the gist of it is in vivo pooled screening that allows you to test multiple interventions in a disease context. I'd love to hear more about the platform, Martin, and I'd also love if you could walk us through those initial conversations with the team. It'd be super interesting to hear how you selected, key data modalities and how to best represent the biology of aging and how it can be modulated.
Yeah, I think that one is sort of easy, right? Like I love to do proteomics and you can't do like 100 million cells. So get tech in, people. Um, so yeah, why are we doing transcriptomics? Like what's the idea there? When we were looking at sort of like what do we want to do with Gordian, there was this core premise of aging happens in vivo. We don't have a cohesive theory of aging, as Eric said. Therefore, we cannot if we make an accelerated, simplified model of aging, we don't know if it captures all the relevant stuff for the specific, like whatever disease we're studying or whatever. And so therefore, you run a risk like by doing anything like that. So can we then instead use natural aging as a model system? Then we know that aging is there, right? And so then it's like a technology review of like, well, could we do that? And if so, how? Like what is the readiness scale of different things? And so at Gordian, we sort of reviewed everything and said, okay, AAV gene therapy is capable of delivering payloads to individual cells within a lot of different tissues of the body, not everything. And at the same time, single cell transcriptomics is like here. And you can see a graph. We had a graph probably in our pitch deck or something. And it's just like number of cells in like sequenced in like the biggest experiment. And it goes exponential roughly, right? With, like Aviv's lab did, I forget. Well, someone did one, right? I forget. And then like Aviv was like 100. And then, eventually 10x genomics and you just go, psh. So that was the combination there. Like this thing is ready enough that, and we think it gives us enough information. So now you don't get all the information about a cell in a transcriptome for sure, right? Like the correlation between protein level and transcriptome is only maybe 50%. And then there's translation, the post-translational modifications and so forth, right? But the way we're thinking about that is like, okay, we're trying to find drugs. And so we're trying to find like, does this thing move a cell towards a healthy state given the perturbation? And so you don't actually need to understand everything. I love to understand things, but like I don't need to understand and have a perfect like picture of everything that happened in that cell to cause that effect. As long as I can tell that it's like healthy, then we can follow up and like understand more later, right? And so the way we think about the transcriptome is not as a like, here's a perfect, you measure everything about the cell and you have a perfect view of what's happening in the cell. It's not that, it can't be, and we don't need that. Instead, it is a relatively unique state of a cell that encapsulates sort of both first order things. If this gene mRNA is up, then there's probably more of it, but not guaranteed. But also second and third order things where it's like when a certain kinase is active because it's been phosphorylated, then these things turn on. And so as long as you're capturing that like state of the cell and you can match that to like which are the directions you want to go in, then you can use the transcriptome as like a phenotypic readout that captures a lot of things, right? And notably, it's like relatively unbiased because when we don't understand everything and when we don't understand how aging things, A, B, C, D, connect to each other. We don't want to just measure like, okay, telomere length, you know, like that's our thing. And like, then we get some result, but it's like, it's underdetermined, like Eric was saying with the clocks.
Yeah, super interesting. So it sounds like it's a very like integrated view. I'm curious, like what types of analyses you're doing. So I think all the chatter last week was probably about like DeepSeek and AI and stuff. And Eric pointed to the importance of AI and generally, computational tools for taking a more integrated view into multidimensional systems like biology. So Martin, how are you using machine learning and these next generation analyses at Gordian?
Yeah, frankly, our focus has always been on like generate the data.
Yeah.
Like I'd rather have a good data set with a lot of perturbations and like well controlled and then do linear regression than, like have some like kind of messy data and then have literally everyone at OpenAI trying to crunch it, right? Which this is not a unintended dig at Retro. I think they're generating big data sets also, right? So because like biology is a lot of dark matter, right? Or at least that's how I think about it. Like there's so much stuff going on that like we don't even know is important yet, right? Like what RNA gets like post-translationally or post-transcriptionally like modified with groups and displayed on the surface of the cell as per sort of like Ryan Flynn, Carolyn Baitosi, right? Like we didn't even know that. And so microRNAs, like fairly new. And so I think the idea that you just like create this sort of like virtual cell and so forth, I think we'll do some things, but how do you predict if we assume, I would assume that the biological behavior is sort of like nonlinear because there's all these feedback loops, right? And so how do you predict nonlinear behavior totally out of your like training set? I'm sure that people at ARC and so forth will like make some cool advances and do some cool things. But it seems eminently possible that we just like get totally constrained by like not have enough causal information to train on. And so I think Gordian for that reason thinks of itself as like a more of an AI enabling company than an AI sort of like pioneering company or whatever. It's like how would you, if you wanted to use AI in drug discovery and you weren't stuck with like this is the way everything is done. Now add AI, right? Like Novartis is like they got the whole thing, and then it's like we need to add AI. Well, then you add AI at each individual step, right? Like you have all your clinical trials, and there's like a lot of documents, and maybe you're like doing things to better understand that. And then you have your like chemists, med chem people, and they're making molecules, and like, oh, can we gen AI some new molecules like in silico? And so you're sort of like doing that at every step, which is different than how if you were just assuming AI, how would you build up your wet lab to empower that, right? And that's sort of more how we're thinking about it.
Gotcha. So it's all about high quality data, perturbation experiments, enabling the AI.
Enabling the AI also for the specific thing you want to solve, right? Because
Yeah.
I think it's like, there's a lot of things that whether you're talking AI or any other sort of like computational approaches, the uses to which we put them is relatively constrained by the data we have. And so if you think about, like Human Genome Project is going to cure all disease. I mean, I don't think the scientists like really thought that directly, right? But it's a thing, a headline that exists, right? And then, okay, let's do the Human Cell Atlas. So just because I think about single cell a lot, right? So we're going to single cell sequence and get the gene expression for all the cells in the whole body, right? And what have we done with the Human Cell Atlas? And I'm actually like, I'm in favor of it. I support that we have like spent whatever it is, $100 million or something like on this effort. But like, what has anyone concluded about disease from like the single cell sequencing everything? To a first approximation, I would say it's like there's different types of cells involved. Like cell types are pretty heterogeneous, right? Maybe there are more subtypes than we thought, something like that, right? Like nobody has actually taken, okay, we have this giant snapshot with like 10 million cells and we measure every gene in every one. And from that, I can say, here is the mechanism of disease, right? Like here's what causes Alzheimer's. Like we don't have the algorithms that solve that kind of question for us. And I think a big part of it is that, like what is Alzheimer's? So there are the data we're generating is like 3 layers of organizational complexity away from the thing that we're trying to predict. Alzheimer's is some syndrome that, like a doctor will prescribe, and it has certain criteria that are like, certainly not super tight, right? Like everyone would say like, oh, is it dementia or Alzheimer's or whatever? It's like a little fuzzy, right? And so you're trying to make predictions around that, and all you're relying on is like a label that this overall brain had Alzheimer's as determined by a doctor, and this overall brain doesn't, right? And so in between, there's like the gene expression comes together as like a neuron. What's the health state of that neuron? How is it connected to the other neurons? Like, what is the flux, right? You're taking a snapshot. So what happens 2 hours from now? What happens 20 years from now, right? And so I think it's like for applying AI, you want to make sure that your data to some degree spans the layers of like sort of conceptual abstraction that you're trying to make predictions across. And if you just go hard on like this thing, like we're just gonna like atomic force microscope DNA and then we'll cure aging. I'm pretty sure that was like a pitch of a company like 20 years ago. But it's like, it doesn't, there are some like question marks in the middle, right? So I think that's like, I'm not like my co-founder knows a lot more about AI than I do, right? But that's my take on it is, do you have the data that actually allows you to apply algorithms, or are you missing stuff?
Now diving back into the biological side, and you mentioned Alzheimer's as one disease that's been researched in depth already. But which disease areas related to aging do you feel present the largest unmet needs? Are there particular conditions or syndromes you think deserve more attention from academia and industry?
So I'll take this one. So I think about it differently. In some way, addressing specific diseases is what biomedical research has been doing so far. And so, huge efforts in terms of Alzheimer's for the last 30 years with very little to show for. One of the things that emerged out of the Buck Institute, Brian Kennedy, Gordon Lidgar, and others, was the whole geroscience hypothesis. And that's a paper that you can look up that is essentially established a whole new way to think about diseases. It came out of the work that was conducted by my Buck colleagues in their own laboratories as they were identifying genes that were making mice or other animals live longer. Also realized that they were not only living longer, but they were living healthier. So there was a really strong effect on health span. And right now, in the human population at this point, average life expectancy is 80, but by the time people reach 65, 70% of people carry at least one of the chronic diseases of aging. So think about many forms of cancer, heart attack, stroke, osteoporosis, macular degeneration, sarcopenia, type 2 diabetes, Alzheimer's, Parkinson's. Let me see, have I gone through the whole list? It's a whole series of disease for which we knew epidemiologically the biggest risk factor is aging. So when individual mutations were identified that targeting aging pathways, they found that animals were somewhat protected, not always, but globally protected against all of these diseases together. And so this led to the geroscience hypothesis, which is that rather than playing whack-a-mole medicine the way traditional medicine does, you treat heart attack as if it were an independent occurrence, you save the patients, and 2 years later that same patient gets a cancer, and 10 years later the patient gets Alzheimer's disease, because they're all driven by the aging process. That's the core factor, the major factor. For example, it's been shown that the aging itself as a risk factor is 7 times more important than your cholesterol level. Cholesterol level is a $20 billion industry with the statins. A third of the American population is on the statin to try to lower their cholesterol. So think about the potential of targeting aging, where you would suppress the risk of heart disease, but we will also suppress all of these other diseases. And that's really the model that we are working on, that rather than attack these diseases when they've occurred, when it's too late, let's go back and try to apply everything we've learned about the science of aging throughout the whole lifespan, and hopefully not make people live forever. taken a very strong stand on the fact that I think extreme longevity, there's nothing right now on the horizon that allows us to make these predictions for humans, but healthy aging, so that most people would actually, you know, live to 90, 95. There's a lot of evidence that we can actually get there. Based on what we know today, I've predicted that most people could live to 95 in good health. That itself is not as glamorous as, you know, walking around and telling people you're going to live to 140 or 180, as some people do, but it is an attainable goal, and one that would be dramatic in terms of society. You know, right now we live on average to 75 in the US, and we spend a big fraction of our lives afflicted by these conditions. Imagine everybody living to 95 in good health, and then having a shorter period of you know, disease that would ultimately lead to death. That's the vision right now, you know, what we are working on in terms of humans. Of course, we are all as excited as everybody else to push the envelope in animal models to really understand what is the aging process. But I hope that answers your question. So in some way, for me, the whole point of disease-related aging in humans is really target the core pathway that is the main driver of all these diseases. And we're making rapid progress in this regard. So I think those therapies might not be as far as we think they will be.
That makes sense. Looking at aging-related diseases to try to see if there's a common pathway or commonality between all of them to understand aging as a whole rather than specific indications. And Martin, do you have thoughts on this as well? Do you agree, or do you have specific diseases or indications in mind that we should
Yeah, I
focus on
mostly
more?
agree, except that I'm maybe more sci-fi idealistic, and I think it's worth aiming for 1,000-year lifespan, whatever the odds are.
Oh,
But
by the way, I'm aiming for this too. I just know that it's going to be a lot harder than we suspect. But I mean, I'm just as ambitious as anybody in the field. I guess maybe I'm a little more of a realist.
yeah. But yeah, I totally agree that like focusing, there's intrinsic limitations to focusing on individual indications, and that's where you know, in a sense, it's like if people get fuzzy about aging, just say effective multimorbidity drug. Like, hey, would you rather have a drug that addresses cardiovascular disease and dementia and arthritis, or just one of those? Okay, well, you'd rather have the first one if you think that that's possible, right? And so the only question is like, can the aging field actually deliver something that works like that, right? Or can a different field slash, depending on where you draw your boundaries, deliver something like that that suddenly has an impact on all these metabolic diseases? And then you can go, you know, like I'm sure Eric is also talking to whatever government and stuff, right? And saying like, wouldn't it be nice if we had like 10 of these instead of like 30 things, of which half are like rare cancers, and you know, like 10 of them are all targeting the same target for one disease, and then there's only 5 actual drugs, right? So like, yeah, if we can do that, it's just more efficient. So then the whole system has not been set up to search for those, right? Like, how are we running the trials? They're for individual indications. And so I think that's where you get into, you know, like Gordian has 4 specific indications we're working on, not necessarily because I think like those, if we cure those, we'll have cured aging, but because I don't want to simultaneously have to innovate on like a lot of science and on like a regulatory pathway, right? So you pick something, and Non-Group has put together like a table with, I forget, like 60 different age-related diseases that if you're spinning out and starting a company and you think you're going to target longevity directly, you're probably not. And you should think about whether maybe you're going to end up targeting one of these things to begin with. And so I think that's like a helpful tool for now. But then at the same time, we should think about how can we change the way that trials are done such that, you know, like you measure more things, and then you can look for biomarkers of specific aging processes, and you sort of weaponize every clinical trial that's done to be something that checks for multimorbidity potential, instead of, you know, like we have like 30,000 drugs and 8,000 diseases, and imagine doing all by all, right? And that's ignoring new drugs. It's just not going to happen in the next 10,000 years or whatever. But you could imagine like, oh, we have 10,000 drugs. If they've all been in trials anyway, can we just like get some extra blood, develop some good biomarkers, and then like test everything for like a potential as aging? Because you think about metformin or rapamycin, whatever you're like have some optimism around, they all came out not as aging drugs. So yeah.
Awesome. Yeah. So yeah, age-related diseases have been really hard to treat. I think the two antibodies approved in Alzheimer's, lecanemab, donanemab, like slow progression by like 30%, but don't really reverse disease. And then there's indications like sarcopenia for which there's no approved treatments. So I think a great takeaway for all of us is that by targeting core pathways of aging, we can make a bigger, more fundamental impact on the progression of disease, actually trying to reverse it, and then also have an impact across different chronic diseases of aging. So I think that's a really powerful message to take away. But yeah, so thank you guys for that portion. And now we're going to go into the question and answer. So I'll start off with a question relating to systems biology. So the question is, do we know if there's a hierarchy to how the human body systems break during aging? And if so, should we build a time stream, so time course of therapies approaches aimed at different life stages to maximize organ resilience?
Maybe I'll just jump in with this one. So one of the sort of initiatives that we have at the back, we recently recruited both Lee Hood, who, if you don't know about him, look him up. He's a giant in the history of medicine and developed the first DNA sequencing machine. So we've hired him and another scientist, Nathan Price. Both of them have been pioneer in the study of the integration of multi-omic data sets, and especially in humans. And I think one of the vision of what we're trying to do is really to try to address the lifespan. So we just got a huge amount of funding from ARPA-H to do exactly the experiment that you were describing. What is the landscape in humans? What happens in terms of those molecular changes? It's clear that when let's say you take a patient that has a heart attack and you measure all of the omics, you're going to find a lot of altered signatures. This pathway is activated and so on. But there's a realization now that what you're looking at the time when the disease manifests itself is the end product of a really complex network dysregulation. Both Lee and Nathan have data originating from a previous company called Arivale that shows that the initial molecular signature of a heart attack can be detected 15 years early than when the heart attack occurs. And so obviously, the vision that we have is that when you identify that initial signature, the dysfunction, one of these genes, by the way, is PCSK9. For those of you who know about PCSK9, it has been somewhat discovered by the whole field of cardiac medicine as one of the most potent treatment to modulate cholesterol. But it turns out to be one of the earliest lesions. Right now in medicine, it's the last thing that you actually, it's the most expensive and the most sort of a top medicine that you're using. But we know it's one of the earliest targets. So think about a world in which the patient would be age 40, and you would detect this early signature. You would be able to intervene in a much more targeted manner, a much more precise manner, and maybe stop the whole cascade that will over 15 years lead to the first network being dysregulated a second. So the whole vision of what we're doing right now with this ARPA-H program that was recently funded with Lee and Nathan Price and other colleagues at the back is really establish sort of I'll call it an atlas of the evolution of system biology over the lifespan. Even more simple, what happens in a 24-hour period? We don't know. I mean, there's incredible biology changes in terms of what we do during a 24-hour period. What happens during menopause in women, which we know is associated with accelerated aging. So we're sort of going back, this is not an intervention. It's just establishing the landscape to understand what is the natural biology of these complex network disturbances. And my prediction is that we will get for many diseases and for the aging process as well. This will allow us to identify much more primary targets versus all the complications. There's a lot of talk about inflammation, for example, as a target. I think it is important, but my prediction is that it could also be a really end of the game target. Inflammation recognizes damage. Of course, when you age, you accumulate all this damage. It's normal that your immune system is activated by all of these. So you can target this, but you also realize that you're only targeting an end product. I think it's going to be much more powerful to go right from the beginning and identify the primary lesions.
Mm-hmm. And this is a super important message for all of us to hear, because it's kind of reshaping the way we think about treating diseases and the way we've thought about medicine historically. And now going back to more preventative early interventions.
Absolutely. If I can make one quick more point, one thing that we've also learned using these phenomics approach. So when I say phenomics, it's the wearables, transcriptomics, metabolomics, proteomics, just microRNA omics, just name it. The wearables, medical records is diseases that we assume to be one condition, like type 2 diabetes is defined when your blood sugar goes from 99 to 101. So that's when you get diabetes. We also know that disease is an aggregate of a whole series of different conditions, probably at least 5. Some that have a more inflammatory component, some that have a mitochondrial component. So the whole idea of these integrated omics, what we call phenomics approach, is really to start really making medicine much more precise at a molecular level. Defining a disease as a blood sugar that jumps from 99 to 101 obviously is a very low resolution type of tool. It works for the clinic, but it doesn't work in terms of understanding what the underlying causes is. So this is the same goes for Alzheimer's. Alzheimer's is a sort of a catch-all term for a whole series of conditions, vascular dementia, some that have more inflammation, some not. So we're conducting all of these studies and all of these diseases, try to increase the resolution. And I think Martin talked about this earlier, this whole idea of precision medicine. And I saw a question on the chat there that was asking, what about precision medicine? This type of phenotypic analysis is going to allow us to do truly precision medicine. And I think that's really the promise for the future.
Thank you. And now a question for Martin. Once you identify hits from your in vivo pooled screening, which delivery systems would you use to deliver target genes? Or would you consider drugging them? And what indications would you focus on? Oh, you're on mute.
Martin, you're muted.
Sorry. Yeah, I answered that in the chat actually. But we have 4 indications that were picked based on sort of like unmet medical need and sort of like reasonable path forward. And then for Gordian, so we have the opposite problem of like a lot of aging companies, right? You do something in like, oh, make my mama live longer, and then I gotta go find out, well, which disease could this work in, right? Like Unity. And for us, it's the opposite. We start with like, we make a choice about an indication, and then we design the whole pipeline of screening and so forth to be optimal for discovery for that indication. So we've already picked our indications from the beginning, but I think that's unusual. And then, yeah, AAV and other modalities.
That's super interesting. Eric, I loved your point about the time course, where it was like in medicine, you have statins prescribed first, and then PCSK9 inhibitors like Repatha or inclisiran. But when you look at the phenotypic analysis and the time course of that, actually you want to inhibit PCSK9 as early as you can. So I think it's great insight. Martin, I'd love to hear what you think about more of that precision approach, maybe as your last or second to last question here.
Yeah.
It's a controversial one because the FDA.
Well, it's not controversial. It just seems really hard, right?
Yeah.
Eric is less daunted by this one. I'm less daunted by mortality than like precision medicine. No, the thing is just like it's tricky. So we need to generate. I mean, this is everything Eric's doing, right? Or the Buck is doing, right? But like you need to first understand what is the general system and then the individual things, right? Like the idea, or at least I am not imaginative enough to like, how do you get to this is your specific cure when we can't cure anything on average, right? And so I think the question is, what are the right studies to do that generate the right balance of data between like the individual variability, and there's different people who are working on this, also like, like Snyder and Brene and at Stanford, for example, right? Like, but then also understanding enough to like put that into a context, right? Because like if you had a truly unique solution to everyone, you would be completely unable to test it without having some kind of like validated biomarker that works for everyone. And so there's sort of like an intrinsic tension in that. But mostly it's something that I am not, I don't feel like I have a great thesis on, like how best to do that. And I'll let other people think about it.
Yeah. All right, maybe one last like quick fire lightning round kind of thing. 1 minute each. Would love to hear predictions for the next 5 to 10 years in this field. Uh, and yeah, we're gonna keep it tight, 1 minute each. Maybe Eric can start.
Well, I predict that in the next 5 years, we'll have the first approved medicine that targets aging. There are a number of phase 3 clinical trials. Actually, Carl Pfleger, if you know his website, it is keeps an incredible encyclopedia of everything going on in the field. And if you look at the number of phase 3 trials that are underway now, we can reasonably predict that one will be successful in the next 5 years. I predict that in some way there's been a lot of complaint about the field that there's not enough funding, and I think it's clear if you look at the potential of this field in comparison to traditional sort of disease-driven type of research, we're going to be transforming medicine. So I would hope that such an event, like the first demonstration that our hypothesis or our way of working is indeed yielding novel ways of targeting disease, is going to open the floodgate of investment, which is both in terms of commercial, but also academic. And in some way, one of the greatest puzzle for me is really, why isn't this field just taking over like crazy? It is slowly. We see an inflection point right now, and I think I want to encourage everyone who's on this call, if you are students and you're considering entering the aging field, I would say you probably will never make a better decision for your scientific life. So I think I'm incredibly optimistic about what's going to happen, but we're being held back in some way by our lack of success. And when that success happens, I think it's going to open the floodgate. That would be my biggest prediction. Next 5 years in this field are going to be absolutely amazing.
I think I would say it's so hard, right? Because you're working on this stuff, right? So you're just like, well, it depends on our will and maybe slightly on like what the US government ends up looking like. Because it's certainly a time of changes, right? I guess I would say serious efforts with publicly available outcomes for developing validated biomarkers. That'll be my sort of prediction, again, partly from just like soapboxing about this constantly and doing work, and Sadvik's doing work on this also. But I feel like that's just such an obvious, like, look, guys, you want anything in like your lifetime, gonna need some biomarkers, right? So I think that's something meaningful. Multiple probably meaningful efforts there will have started with different philosophies. Some people are going to be like, we're going to find the one measure of aging. Some people are going to be like, we should just take these specific mechanisms and make predictors there. And then others may be like more in the personalized way. But, like go beyond, like we all have a clock that's like sort of unvalidated from some vendor, and everyone's doing different vendors, and some people have an Apple Watch, and there's a lot of data, but it's very disparate and not rigorously tested. And then if that happens, right, like then things will speed up. And then I'm hopeful for what Eric says as well. Although I think the pipeline is not like super thick of things, because like if it gets approved in that period of time, it has to like be in trials already. So it's just a few things.
Yeah, that's totally fair.
There's at least five phase 3 trials right now on drugs targeting. So 5 phase 3, that means probably, you can expect, I would predict 2 in the next 5 years will be approved. And that really, I think, could be transformative. And I agree with Martin in terms of really, I'm part of a group called the Biomarkers of Aging Consortium with Vadim Gladyshev at Harvard and Mike Snyder at Stanford and several David Furman and several other colleagues, Nathan Price at the Buck. And I think this group has really, I think, done important work in terms of codifying and really sort of defining what is a biomarker of aging. We have a whole series of papers that I invite you to go read if you're interested. Sort of defining the problem and then hopefully, moving forward. There is also a new government initiative that you might have heard about called Prosper, which is going to be unleashing, we hope, a large amount of funding to really define what is an effective and actually portable, because it's good to have a $5,000 proteomic screen, but it doesn't really help the clinic. So how do we translate this into something that you could actually do at home? Last but not least, for me, one of the areas that I'm truly excited about is the wearables. They are sort of growing by leaps and bounds, and I think I could see a day in the future where they will become a primary tool of your health, using multifunctionality that goes way beyond CGM and HRV and all of this, but really integrating all of this data. I think there's incredible potential there. Some of them now measuring glucose without any invasive techniques. And you can imagine a lot of variables eventually being measured in this way, along with your blood pressure, everything else. So that's going to be really important in the future.
Super interesting. Well, thank you both so much. I'm going to turn it over to Sean to close out real quick.
Thank you very much, Sadvik. Well, it has been an absolute pleasure to hear from both of these visionaries in the field. All of you got the opportunity to hear on so many different topics that are going to be explored more in depth as we continue to go throughout the panels in the next several months. So first and foremost, I just want to say, Martin, Eric, thank you so much for your time again, and thank you for the work that you're doing for the field. It really does mean a lot to all of us. Now, to everyone in the audience, definitely be sure to stay tuned. As I mentioned earlier, we will be posting on the Nucleate socials as well as on our Luma Global Events calendar the information for the future panels taking place. The next one's going to be happening on March 3rd, talking about a path to the clinic for aging. So some of the thoughts you just heard at the end there about some of the phase 3 clinical trials, those will be discussed in more depth on March 3rd. So definitely be sure to keep an eye out for that and join us there. We'll also be announcing soon our resource guide with plenty of links and information covering so many of the different topics that you heard touched upon today. Thank you to all the audience members who tossed their questions in the chat. I know we didn't get the chance to get to every single one of them, but we really appreciate it, and we really encourage you to stay engaged. We hope that this is just the first of a great series focusing on the future of aging biology, and we thank you so much to all of you for joining us. Thank you again, and we'll see you at the next one.
Good to see you all.
Thanks, Eric.